So, thank you so much. In 2012, I was coming hot off of my early career grant. I flew to DC, and I walked into the director of photography's office. And I pitched a story about this creature. This is not a photograph I took. This is from a scientist at the University of Nebraska. But this is the kind of images that I would find online when I was researching this project. So you can imagine the skepticism of Kurt Muchler. He was the director of photography. As he looked at me with kind of a squint, and he said, "How are you going to photograph this?" And the truth is, at that time, I had no idea how I was going to photograph this. But I got a lucky break, which is that National Geographic already wanted to do a story about this. This is a parasitic worm that infects a cricket. And so they were willing to take a chance on me and let me figure this out. So here's my first attempt at photographing this interaction. And I think this is a decent photograph. You can see the details. It's an improvement over that image I saw the scientist take, but it wasn't interesting enough. It didn't show what I wanted to show clearly enough. So I tried focusing on the worm. And okay, you can see the worm, but it's a little weird. There's a random cricket leg sticking out of nowhere. I tried getting a little artsy and weird, and I think I got a little too artsy and weird. Like this, you can't tell anything what's going on here. I tried a more straightforward shot from above, and this, again, was not good enough for National Geographic Magazine. So in total, I spent about 28 days. It took about 3,000 images for me to get one that was worthy of the magazine. And I actually had a friend film my process of getting that final photo. So here I am with a tank full of infected crickets that has been shipped from New Mexico. I'm storing them in my kitchen in Berkeley. And every day, I would pick out these infected crickets, and I would look for the ones where this parasitic worm is ready to emerge. And you can tell because their bodies darken inside the belly of this cricket. You can see when they're ready to pop out. Then I set up my lights in a makeshift studio in my backyard. I probably should have gotten a haircut before I asked my friend to film me. And then I'd set my lights. And so I would build a little tray and then fill it with this special liquid called cricket saline. This is a mixture of salts that mimics the internal chemistry of crickets. And I'd put the crickets in the refrigerator that would slow them down, and this is all to trick this parasitic worm into emerging from its host more slowly. And because that worm is cold and it's emerging into an environment that it thinks is still inside the cricket, I had enough time to get my final shot. So what's happening here, this is a horsehair worm that infects crickets, and it grows up inside their body. When it's ready to come out, it needs to find water. It's aquatic as an adult. And so to find water, it forces the cricket to find a stream or puddle, and it makes that cricket drown itself so that it can safely emerge. This is a story about parasites that have the power to control the minds of their host. And my job from the very beginning was to find a way to make compelling images of a viscerally uncomfortable and unpleasant subject. And so I'm going to show a few images from this story as well as a couple of others I photographed for National Geographic. Here's another parasite that takes control over the mind and body of its host. This is actually a ladybug standing guard over the parasitic cocoon of a wasp. That wasp injects a single egg into that ladybug's body. That baby wasp hatches inside, and it feeds on the inside of the ladybug without killing it. It then emerges from its body, it spins a cocoon, and it forces that ladybug to stand guard over it as its protector. And this is an image that National Geographic decided to put on the cover of the November 2014 issue, which is an honor not given to many bugs these days. And if you look at this, you might be wondering, okay, this doesn't look like a normal nature photograph. And that's intentional. I was trying to find a way to connect to people who wouldn't normally care about parasites. And I was drawing on 3 different aesthetic influences for inspiration. Those 3 things are film noir, graphic novels, and Japanese animation. If you're wondering what Sailor Moon has to do with mind-controlling parasites, let me go over that again. Hard lighting, dramatic backgrounds, and selective attention to detail. The key insight of anime artists is they recognized they didn't need to accurately represent every detail in their subjects, in their scenes. Certain details carried more power than others. So when you look at her eyes, they're way out of proportion. And they have all these colors and reflections inside. And they put way more attention into her eyes than the texture of her skin or the texture of her hair, because they knew her eyes could convey so much more emotion and energy. So let me show you a couple more examples to illustrate how I applied this idea. This is my favorite example of a host-manipulating, mind-controlling parasite. It's a sheep crab that's been infected by a parasitic barnacle. And this parasite is tiny, it's barely visible to your eye. It crawls inside of the body of that crab. If that crab is male, it turns it into a female. The parasite gives its host a sex change, meaning these feminized crabs grow an egg chamber that only the females have. And the parasite lays its own eggs inside, and it activates the maternal care instinct of the parasitized crab so that the feminized crab cares for those parasite eggs as if they were its own. So what you see here, all these tiny little specks, those are tens of thousands of parasitic barnacle larvae emerging from this crab, ready to infect more crabs. And I'm very carefully thinking about where I'm putting light and where I'm keeping it off, because I don't care about any of the details of this crab. That's not an important part of my story. I want your eye to be drawn to these tiny little specks in the back. And actually, as a scientific description of this interaction, this would not be that useful. This is not the kind of image that I would submit to a journal describing this behavior. That's because my purpose in this image is different than capturing every detail in a way that could compare this to another crab or to another parasite. It's more about stopping the reader of National Geographic and inspiring them to read the caption, to read more about this story. This is a frog that's been forced to grow extra legs by a flatworm parasite. The flatworm deforms the frog in order to turn it into bait, so it's more likely to get eaten by a heron. That's because the parasite is just using this frog as an intermediary host, as transportation to get into the gut of a heron, which is where it can complete its life cycle. Each of these images I took in a different place, and for each of these places I would set up a second camera pointing back at myself that would take a time lapse to show my process. So for example, to show my process, hotel room in Costa Rica. Try to go back there. This is a hotel room in Costa Rica, where I photographed a spider, and I would keep these spiders in these white tubs in the corner, and I'd have to hide them from the housekeeping staff so they wouldn't get thrown out. This is another hotel room in South Dakota. You can see that most of my job is actually just rearranging hotel room furniture. Sometimes I get lucky, I get to work in research labs. This is a lab in Montreal where Fanny Moray was studying that ladybug and its interaction with that parasitic wasp. And this is a lab where I photographed the deformed frog at the University of Colorado Boulder. So midway through this project, I had to send an update to my editor to show the best images I'd taken. And I was behind my deadline for this, and I was staying up late, scrolling through my images, and this funny thing happened. It was about 2 o'clock in the morning. I was listening to techno music to keep myself awake. And the patterns of the images and the patterns, the the beats of the music started to synchronize. So as a joke, I made a stop-motion video that showed all of my images in order, set to this music. So I'm going to show you a shortened version of the dubstep Parasite music video that I made for National Geographic. Here you go. Thank you, thank you. My next story for National Geographic is about a creature that may be more familiar to you. So this is a honeybee. And if you've been reading the news, you may be familiar with the fact that over the last 10 years, they've been having a pretty rough time, especially in North America and Europe. But to be honest, when National Geographic asked me to photograph this story, I did not want to do it. That's because I felt like this story had already been told. It had been told dozens, hundreds of times by other photographers, other journalists. I wasn't sure what new thing I could bring to this story. But I also didn't feel like I was in a position to say no to National Geographic. So I decided I had to try anyway. And the way that I started is I tried to take all of the lighting techniques I developed for parasites and apply them to bees. Again, it's not a bad photograph, but to me it wasn't compelling enough to inspire somebody to read this story. It didn't feel like it was surprising, it didn't feel like it was new. And I spent months trying to figure out new ways to photograph these creatures, and I would take close-ups of their face, I would photograph bees inside of almond flowers. And I think they were perfectly fine photographs, but they weren't new. And about 8 months into this story, I got a message from my editor that said, you know what, we're kind of concerned about your progress on this story, and we really need to see a new direction from you, ideally in the next week. And so I'm kind of freaking out now. You know, I was happy my first story made it on the cover of National Geographic, and now I'm about to blow it. And I've spent months trying to photograph these creatures, and how am I supposed to figure out a new way to do this in a week? So I went to Davis. There's a bee lab at UC Davis in California, and I would work with the scientists there, and they would lend me frames of beehives, a part of the beehive that I could take home, and I would stare at this thing, trying to figure out what can I do that's new. And I saw this bee that was emerging from its brood cell, this little chamber inside the comb, and it had gotten stuck there, and its furry little head was just sticking out. And I had this idea for how to show the bees not isolated from their environment, but within the context of where they live. And this became the opening image for that story. And this was the story, this is the image that also convinced National Geographic to give me more time to figure out this project. So this is a newly emerged bee inside of its comb. And I realized this story was not about just taking close-up portraits of bees, but it was what I wanted to do was to show how scientists study this creature and the experiments that they're using to try to understand the threats that they're faced with now. So I went to Louisiana State and I photographed this experiment that Frank Rinkovich did to try to understand the effects of pesticides on bees. He'd put the bees in a little paper cup, sedate them with carbon dioxide, and then drop a tiny bit of pesticide on them to track how it affects their health. This is an experiment at Chris Mullen's lab at Penn State, where they would train bees, they would stick them in these little plastic cones, expose them to a little bit of floral scent, and then give them a Q-tip dipped in sugar. And if they repeated this a few minutes later, give them that smell, and they see that the bee sticks its tongue out, they know that the bee has learned to associate the smell with the reward. It's a way to get a rough idea of the memory ability of bees. And they can look at some of the sublethal effects of pesticides or other agricultural ingredients to show that there are chemicals that don't kill the bees outright, but disrupt their ability to remember where their food is. Does anybody know what's going on here? This is an image from the USDA of a really critical tool in bee research. And I saw this, and I knew I needed to photograph it, but I didn't know how I was going to do it. In the end, this is my take on what's happening. This is the artificial insemination of a honeybee. This was done at another USDA bee lab. This is Sharon O'Brien's hands, the lab technician. And actually, my photograph doesn't show what's happening any more clearly than the previous one. But again, that's not my goal. My goal is not for somebody to be able to recreate this process by looking at this image. My goal is to spark a question. Namely, what the heck is going on here? And the reason that this is so important, this is a way that scientists can actually breed bees in a lab and select for traits that make them more resilient to parasites and other diseases. And for this story, I actually started keeping bees in my backyard as a way to research them, a way to understand them better. And I would see all the different stages of their life cycle. I wanted to show that entire process of metamorphosis that I knew was happening in front of me, but that it was happening too slowly for me to see. So working with Billy Sink at UC Davis, we figured out how to raise bees in front of a time-lapse camera. I'm going to show you the first 21 days of a bee's life condensed into 60 seconds. This is a bee egg as it's hatching into a larva. Those larvae then swim around their little chambers, their brood cells, feeding on this baby food that other bees secrete for them. This is the pupation process. This is where the larva turns into a pupa. Its legs and its head start to separate. This is the same pupation process seen from above, and you can actually see the parasitic mites running around in there. Then the tissue, the cells reorganize in their head, and the color develops in their eyes. The last step of the process is their skin shrivels up, and then they sprout hair. So the first story that I pitched National Geographic, before I even mentioned parasites, was on hummingbirds. And when I proposed hummingbirds, and I was describing what I wanted to photograph, the editor cut me off and said, "That is a boring idea." Because every photograph of a hummingbird that can be taken has already been taken. And she was referring to photographs like the one I took here in 2009 of an Allen's hummingbird at Berkeley. And this is a perfectly nice photograph. It would make a great postcard or calendar. But there are no shortage of photographs like this one. And so my goal here was to take a universally beloved creature and find a new way to visualize it. This is, in some ways, a lot different than the challenge I had with parasites, which was taking an ugly thing and making it look interesting. Here I'm taking a beautiful thing and figuring out how to reimagine it. And my approach here was to look at hummingbirds through the eyes of scientists. So for example, this is a hummingbird as it's drying itself off. And this is a phenomenon that was only recently described by Victor Ortega-Jimenez and Robert Dudley at UC Berkeley. It's essentially the same process that happens with your dog, but the hummingbird is doing it many times faster. It's shaking itself so vigorously that water droplets are shooting off of its body. And this is how scientists study hummingbird aerodynamics. They look at the airflow around a hummingbird's wings by using miniature fog machines. And this is how a hummingbird tongue works. This is work done by Alejandro Rico Guevara, who figured out that they have squishy tubes for their tongue, a forked tongue. And as that squishy tube gets compressed by the bill, and then it touches nectar, it springs back to its original shape. And that acts like a little micro pump to draw in nectar up to 15 times a second. And these are the videos that inspired this project, all about hummingbird biomechanics. And when I saw these videos, I felt like this is groundbreaking work that is not getting the attention that it deserves. And so my goal was to figure out a way to show this research in a way that would capture people's attention who wouldn't necessarily be interested in it in the first place. And I think that is the key value of storytelling. Its ability to connect people to what you care about. So the last video I'm going to share with you is my recreation of these experiments to show the amazing biology of hummingbirds. Here you go. Thank you.